describe how meiosis happens as a process of cell division. Again, we have to start with interphase. Again, interphase has 3 parts, and the most significant is synthesis. During synthesis, it's going to double. And if this is the cell that I start with, let's say I've got 4 chromosomes. These 2 represent one from my biological dad, one from my biological mom. They're homologous to each other because they both have genes that code for certain things. They both code for eye color. They both code for liver function. They both code for, say, heart disease. These 2 are homologous to each other because they both code for tongue rolling or for the propensity to get arthritis, for instance. During interphase, they're going to synthesize and make more. So during interphase, I'm going to represent that with having now 2 of each, right? I've already started to change during meiosis. I've got 2 of each. And during prophase, not only do they become present and visible, but we've also got this phenomenon known as crossover happening during prophase 1 of meiosis. So we're going to represent that with different colored yarn here. When crossover happens is right after synapsis. So the homologous pairs are going to synapse together, and now they're in close proximity, and that's when they can have a little crossover happening. So this gene would jump from one to the other, and so we're going to represent that with the yarn jumping over. So now I've got the crossover happening, and I've got genetic variety. We're going to show crossover happening for these 2 genes changing over. So I'm going to take away that one and take away that one. We would show crossover happening with our yarn, genetic variety. Now, when we move to metaphase, these homologous pairs are going to line up, and that's important to remember that they're still in this homologous pair arrangement. My homologous pairs are going to line up in metaphase 1, and then in anaphase 1, my homologous pairs are going to come apart. And then in telophase 1, I've got 2 new cells that are going to create a new nuclear membrane around them. We're going to keep those 2 new cells after cytokinesis and come down here for prophase 2. In prophase 2, I've got 2 cells, so you're going to notice that in slides where it's 1 unit, but there are 2 cells involved already, and they're starting to be present, but I don't need to go through crossover again. In metaphase 2, we're going to line up along the midplate again, so all these are going to line up. And 2 cells. In anaphase now, this is key, we've got to have the sister chromatids coming apart. So this is going to come apart, these 2 are going to come apart. This is where independent assortment happens because we're not sure which one is going to go with which. This could have easily gone with that one instead. And that independent assortment also leads to genetic variety. And so this is all happening in 2 separate cells already. So now at the end of telophase 2, when that cleavage furrow happens and the cytokinesis is the pinching of the cytoplasm, now I've got 4 very different looking cells from what I started with. This is what my parent cell looked like. And now to finish, I've got 4 very different looking cells, right? I've either gone through spermatogenesis to create 4 new sperm that look very different from my parent cell, the spermatocyte, or I've got 3 polar bodies that will be absorbed again and 1 egg. The parent cell was diploid or 2N, and each of these daughter cells is only 1N. It's just half the amount of chromosomes that the parent cell had.